welcome to our Aging Studies Research Group online seminar. The Aging Studies is one of the research groups within Brunel University, and our focus is on aging, but we very much do it from a perspective of many different disciplines, interdisciplinary. So we go across the whole university. There are members of our Aging Studies group, and hopefully today you'll have a taster of some of our the range of different research and the different novel and interdisciplinary research we do today. So we welcome you all to this series of presentations. To let you know some admin stuff, there's going to be each presenter is going to be speaking for 15 minutes, maximum 20 minutes, and then there'll be up to 5 minutes for questions afterwards. So after each presenter, you have the opportunity to ask any questions of interest that may be of you. So please, if in the meantime you have any questions, please pop them in the chat, and we'll ask them at the end of it. We'll look at them and put them in. So if you think of any questions that's going along, pop them in the chat, and we can ask them as we go along. Towards the end, we can ask them as well. Unfortunately, Professor Christina Victor can't be here today. She might drop in later in the meeting, but so just to say that I'm sorry, she won't be presenting today, but we still have 3 papers. We're going to first have Dr. Elmer Kahl, who will be talking about freezing of gait and Parkinson's. We've got Professor Joanna Bridger second, doing snails of men. And then Ni Qing about Chinese migrants and low carbon lifestyle. So I suppose without further ado, Elmar, do you want to share your slides? And welcome, and we look forward to your presentation. A next step, co-creating new interventions targeting freezing of gait in people with Parkinson's. And if you want to say something about your discipline as well as you start, that's great too. Thank you.
Yeah, of course. I'm going to share my slides first. Let's see if it all works. 1 second. There we go. Is this visible for everyone? I think hopefully it is.
Yes.
Yeah, okay.
That's
okay.
Great. Thank you.
Yeah, so I'm Elmar Kahl. I'm a lecturer in physiotherapy. I'm not a physiotherapist myself. I'm a movement scientist, and my broader background is looking. I'm interested in studying how psychological influences, so things like anxiety and changes in attention, how they can influence how we perform movements, so how we walk, but also how we learn or relearn movements that movement abilities we may have lost. So after stroke, for instance, I've done studies on how we can help people relearn balance skills again using different types of instructions and learning methods. But part of my research is also concerned with people with Parkinson's, and that's what I'm going to talk about today. And I'm going to talk about how we, the title of this meeting is New Perspectives on Aging. So in this case, I'm going to talk about how we try to integrate the perspective of people with Parkinson's themselves in the development of new interventions to help them with freezing of gait. So I'm going to explain what that is in a second. So yeah, let me get started. There we go. So what is Parkinson's? Let's start with that. Well, some of you might know this, but some might not. So I think it's important to clarify. So Parkinson's is a neurodegenerative disease that affects a lot of people in the UK. And in fact, it's the fastest growing neurological condition in Western countries. And it involves progressive damage, so worsening damage to specific parts of the brain. And typically this starts with deeper lying areas, subcortical areas, we call them, which are very important for the voluntary control of movement and sequencing of movements. And you can therefore expect a lot of motor symptoms, so motor issues to be evident in people with Parkinson's. So you may know that people often have tremor, but also difficulty with balance and gait. And freezing of gait, which I'll come to in a second, is one of those symptoms. However, when the disease progresses, so over the years, usually, what you can see is that also areas that are more frontal, that more at the front of the brain, get involved as well. And at that stage, you can see that cognitive issues start to emerge. So issues related to the planning of actions related to inhibition also from maybe behaviors you don't want to show. Inhibiting those behaviors can be more difficult. And affective issues are also evident. So that's to say that people sometimes get worsening anxiety symptoms, more difficulty with regulating their emotions, etc. So actually, Parkinson's is quite a complex disease that's definitely not only a movement-related disorder. So I summarized some of these motor, cognitive, and emotional affective symptoms here on this slide, just to show you that those motor symptoms like tremor, slowness of movement, etc., but also cognitive issues like issues with attention and memory, and issues related to psychological functioning and well-being, anxiety, depression, that these all basically interact. So if you have issues with anxiety, you feel anxious, you feel fearful, this might result also in changes in how you move. Similarly, when you're very anxious, this might also influence how you direct attention and your ability to do so effectively when you need to do a task like walking. So the interplay of these three things, how these interact, that is actually where freezing of gait comes into action. So freezing of gait, I'm going to show you an example to exemplify what this is. You can see it in this short video, hopefully. Sometimes it's a bit more jerky on Zoom, but what you can see is a person that tries to walk to the bed at the front of the screen, but is actually taking very small shuffling steps and sometimes having a complete halt like here, where it actually is temporarily for a little while impossible to really get the feet from the floor well. So freezing is typically called or described by people as having feet stuck to the floor. So what we sometimes do is actually give people certain cues to help them walk more effectively. You can see that here. This person now gets a ball on a leash. And interestingly enough, it's the same person, just a few seconds after not being able to move at all, you can see a much more fluent walking pattern. So we actually have ways to improve freezing of gait and to help people like get out of such events. But such cueing strategies don't work for everyone and don't work always for the same people. So we are always looking for a better understanding of what freezing is, what it is caused by, to help inform better treatments. So what are the causes? Well, that is actually not fully understood. So we know something about what the biomechanical causes are. So for instance, what you can see here is just a sort of summary of what we do when we take a step. And what you need to do, so when you want to get out of a freeze and want to take a step, is we need to shift our weight. It sounds simple. So basically what you want to do is when you're standing with your feet together, you want to have a short weight shift to the right foot in this case, and then a bigger one to the left and forwards. So doing so would help you get your right foot off the ground, because by moving to the left, automatically you will take your foot off the ground. And then by moving forward again, you would then take that step with your right foot. And what you see in people with Parkinson's is that this weight shift, and you can see that sort of simplified, presented here in a simplified way. This red line here is much less directed, it's much less effective. And basically what you see is that people with Parkinson's find it really difficult to make this specific and sufficiently large weight shift in order to take that step. So we need to make sure that cues that we use, interventions that we use, can address this and can help people make this weight shift, but also in a safe way. You don't want them to make too large a weight shift, because then you can fall over, of course. So this we know, but interestingly, there's also something else that plays a big role in freezing. So what we see is that certain psychological factors also are important, and particularly anxiety. So I was just telling you that some issues with anxiety and fear are actually quite common in people with Parkinson's. And researchers have shown that these people have a higher risk of also experiencing this freezing of gait. So for instance, in a study by Martens et al. Around 10 years ago, they let people with Parkinson's who experience freezing in daily life, they let them walk in an environment where they had low anxiety, where there was just walking on a solid surface at ground level. But then they also put them in a virtual reality environment. So they fitted them with glasses that simulated a high environment. So an environment where they were walking at height with like a cliff edge on their sides. So that induces a lot of anxiety. And when they did this, you can see that the time spent in a frozen state, so the time spent in a freeze, increased about 2 to 2.5 times. So you can see that suddenly 10% of the time walking was spent frozen when they were not anxious, but when people were anxious, it was around 25% of the time that they were walking. They were actually in a freeze, in a freeze. They couldn't get their feet off the floor. What we don't really know is why anxiety does this. So what happens? What makes it that anxiety results in people having worsened freezing? Because if we understand this better, we can actually target freezing and help come up with interventions to help address this and make freezing less severe in people with Parkinson's. So what we decided to do, and this is where the new perspective comes into play, is actually start with interviewing people with Parkinson's and ask them exactly about what their experiences are when they have a freeze, and especially when they have memorable freeze that really have a lasting impact on it, that really they can recall even a long time later. So what we've done is we got 35 people with Parkinson's into Brunel, and we sat down with them and interviewed them on their experiences to really understand their perspective on this issue. And particularly, we asked them about what they thought the factors were that would help them get out of a freeze, so decrease freezing severity, but also the factors that actually contributed to worsening of this freezing. So we did a thematic analysis that was led by my colleague, Dr. Mariel Norris. And in that analysis, we also involved people like myself with an interest and expertise in motor learning and Parkinson's, and other clinical academics to interpret what people have said and how that fits with our understanding of freezing. And this resulted in a new perspective on what causes freezing. So what does it look like? Well, this is quite complex, so let me talk you through it. So first, what we found, and that you can see in the highlighted bar here, is that people talk about freezing as there being different levels of freeze. So some people say that there are instances where they experience a deep freeze. So you can see that to this side of this figure, where they really have a very severe, very invasive freezing event that it's really hard to get out of. But there's also mid-light freezes, and even, of course, free movement where you don't have a freeze. So people describe this as being on a continuum rather than a freeze or not a freeze. People have experiences of this being of different levels of intensity. And what helps to actually move from a deep freeze to lighter freezes or even free movement is focusing attention on movement. So people really commented on coming up with strategies that helps them to, when they're in that freeze, to really start thinking of, okay, what do I need to do to get out of this? How can I help myself to make a step again and get out of this phase? Problematically, though, freezing often occurs with anxiety, and this anxiety seems to actually work against getting out of a freeze because it distracts people from their paying attention to what they need to do. So very many of the people in this study actually really commented on fear and anxiety related to freezing and what freezing signifies to that, resulting in distracting thoughts and ruminations that then prohibit them from really focusing on what they need to do. And this is also in relation to more accumulated experiences that people have when they have multiple freezes over time. So typically people have freezing events every now and then. So usually some people only have it like once every few days, but other people have an increasing frequency of freezing, especially over the years. So as the disease progresses, typically you see that freezing gets worse and worse. And people know that this is a marker also of progression of their disease, so worsening of their condition. So often this leads to feelings of vulnerability, but also a sense that they are not in control over their condition and that it's getting worse. And this, of course, reinforces those worrying thoughts whenever they then encounter another freeze, making it even more difficult to get out of a freeze. So over the years, you can see that any freezing event really adds up to sort of an accumulated sense of a loss of self and a loss of control over one's condition. And as I say, that makes freezing even worse. So what we really found in this model was that there's a sort of interplay between being able to focus your attention on your movement to help you get out of a freeze, but also anxiety, on the other hand, making this increasingly difficult. So this is very important to understand, because that means that when we want to target this and come up with interventions to address freezing, that we need to come up with instructions and strategies that help people sort of block out that anxiety and really redirect their attention to their movement. Okay, so how do we use this now, this knowledge? How can we sort of harness this to help develop interventions to step from a freeze? Well, we know that anxiety results in freezing, and that anxiety may worsen freezing through distraction and through worrisome thoughts that people may experience. So we need to come up with cueing strategies that help maintain focus on movement, even when this happens. What we've done, we therefore summarize this in, and I'm going to talk about that intervention, we try to use this information in a particular intervention where we combined explicit instructions that direct attention to the required movement, so to that weight shift that I was talking about at the start. So coming up with instructions really that help people to move their weight on one side, then to the other side, and then forwards to help make that step. We combine that with a dance cue that requires rhythmic strutting of the hips and that hopefully is easy to recall, that creates a really vivid image that people can imagine whenever they are in a freeze, because that's the difficulty. When you're anxious, when you're fearful, and you get distracting, worrisome thoughts, it gets increasingly difficult to recall those instructions you got from your therapist. But if you've practiced something that stands out a little bit, that's a bit different, but that does result in the weight shift you need to make, and that is also easier to recall, even when you're anxious, then it could have really meaningful and powerful effects on your ability to step from a freeze. So that's what we tried to do here. And I must say, I can't move my hips that way, but that is roughly the image we wanted people to have in their head, so that they knew, okay, this is what I now need to do. I know I'm in a freeze. It's not nice, but I can get out of it if I have this strategy and I use it. So this is a paper we published last year on a feasibility study where we tried this out, where we basically use these perspectives of these people with Parkinson's to then inform the development of this intervention and test it out. So we use this dance cue to help train this group of people with Parkinson's and see how it helped them or not helped them to get out of a freeze, see if it had an effect on that. So you can see some information on the people with Parkinson's here. On average, they were 70 years old, but they ranged between 56 and 83, with an even split between male and female participants. And on average, they had their diagnosis of Parkinson's around 10 years, but again, it was a wide range. I won't bother you with those details down there about the Hoehn-Yahr UPDRS scores, but they basically showed us that people had some impairments. On average, they were moderately affected by their Parkinson's. So what we've done with these people, we created an intervention related to this cha-cha cue. But first, before we let them practice this, we did a baseline assessment. So we wanted to see when people were turning back and forth on the spot, which is a particular movement when people often experience freezing. So when they were doing this, how often would they freeze? That was basically what we were interested in, and how well would they be able to get out of the freeze using the strategy they would normally use. So before getting training with this dance cue. Then we let people practice with the cha-cha cue, listening to music, and in front of a mirror, we let them really experience making this weight shift in a rhythmical fashion. So only when practicing, they got some visual feedback to really help them internalize this image of what that movement should feel like, so that whenever they then would be retested, whenever they would be in a freeze, we would then ask them to recall this image and try to move in that way to help sway themselves out of this freeze again. So this was a really fun study. Was actually, the intervention was led by 2 MSc physiotherapy students who were professional dancers. People really seemed to like this intervention. And after this intervention, we then let people again do this turning assessment back and forth. But now, whenever they would encounter a freeze, we asked them, okay, when this happens, if this happens, use the strategy you just practiced, try to recall that image and try to get out of the freeze in that way. Okay, so one thing to clarify, I'm going to show you a video of what this looked like. People are wearing a headset because we had them always making these turns at the top of a flight of stairs. This was deliberate because we wanted to make sure that people actually froze in the lab. And in order to do that, you have to make people a little bit anxious. So that was the idea of that. So just so you know. So what did we find? Well, we found a lot of freezing episodes during baseline and slightly fewer during the post-test. So after training, people had fewer freezing episodes. 10 people had freezing both at baseline and during the post-test. And what we found is that people were more successful at getting out of a freeze, as making that step, after having trained with the cha-cha cue, with the dance cue. So you can see here that the success with making a step out of a freeze was 20% higher, was twice as high, basically, when people had practiced. So 40% compared to when people hadn't practiced. So we saw a big increase in success of people to, whenever they did freeze, to get out of this freeze, which was quite useful. It was a good result. And here you can see what it looked like. So here you can see a participant. This is all with permission, by the way. Here you can see a participant trying to make a turn, but actually being stuck in the moment here and really struggling to now finish this turn and make a 180 turn. So there we go. Another freeze. You can see some tapping on the leg. So that's apparently the strategy they used themselves. But it wasn't really subtle, and it took a long time to really take that step and complete the turn. Now, what I hope you can see here, but I can post links to the videos later as well, is you can see a more subtle turn. No freezing happening here. Now, when turning left, there's a small freeze, but also a gentle sway of the hips, getting out of the freeze again, and another sway of the hips and completing the turn. So yes, this person still froze, but it was easier to now complete the turn with a minimal delay and only with a short duration of a freeze. So we saw, and this is exemplary for the results for the group, we saw clear improvements here in turning behavior. Okay, so if you now recall biomechanically what I was talking about with what is required to make a proper weight shift and initiate a step, if you look at what happens with people's sway, so how much they were swaying around when at baseline versus after practice, you can also see that at baseline, the sway was quite irregular and all over the place, basically. So no clear sway to the right, to the left and forwards. But at post-test, when we looked at how people were swaying, you can see that this shape here really resembled this ideal shape that I showed you at the start. So also on that level, we saw clear improvements. So what does this mean? Well, this was a proof of principle study. We found that using this new information from people with Parkinson's themselves, using their experience to inform our understanding and then inform an intervention, led to really promising results. But now we need to actually, and this is what we're planning, to do a bigger study where we include a control group, where we have more methodological rigor in place to really understand if this intervention really works also in real life. And this is what we're trying to do now. We're actually co-creating videos that teach people with Parkinson's, that they can use at home, basically, to teach them how to mitigate the effects of their anxiety and how to create this weight shift when being stuck in a freeze. And the idea is that they can use this independently at home, and that we're then going to see when they are in their home environments, but also in their local community, whenever they encounter a freeze in daily life in such environments, whether they then can indeed, as in the lab, effectively get out of the freeze in a safe way, but also in a more successful way. So the intervention that we're co-creating at the moment includes instructional videos on the management of anxiety, but also videos with specific instructions on how to promote a weight shift while also maintaining safety. So as part of this project, which is led by Will Young of Exeter, we're also doing a very large public involvement study where we're actually also evaluating how people with Parkinson's themselves experience this process of being involved of developing these interventions, and if they feel to what extent their input has really changed the interventions and the study we're running. So that is actually a very important component of the project we're doing. Yeah, so this is a summary of what we're trying to do, and the idea is that all this work should be led and is led in our perspective, at least, by people that it is meant to be used by. And we really are keen to use this model of working more often in other applications in aging research as well. So yeah, that was in a nutshell what I wanted to share. I hope I was on time.
Yeah, no, that was great.
I tend to talk
Thanks,
too much.
That was really, really interesting. Now we're going on to our second presentation by Professor Joanna Bridger. I'll let you say a little bit about your discipline as well, because as we're doing a range of different disciplines, I think, Janet, you're going to share the presentation for this one. So we're going to have a few of the next slides now. So we're used to hearing those on these Zoom calls. Welcome, Joanna, of Snails and Man, and I'll let you say more about your discipline. Thank you very much.
Thank you. Yes, I'm sorry that I'm not directing my own slides. They're not functioning very well in Zoom, so it's probably my fault. So I'm in biosciences here at Brunel University, London, and I've been here at Brunel 23.5 years. So I really quite like it, and I decided to stay a long time, so, and hopefully even longer. But I'm going to talk today around aspects of the cell genome. I'm interested in how the genome functions, and it's really the coming together of two different interests, or two different projects, long-term projects that have been running, that have sort of come and collided together. And I want to talk to you today about these snails, which are a freshwater snail from you find in the tropics, that are actually a host for a quite serious parasitic disease called schistosomiasis, bilharzia. And so I used to work on this a very long time ago, and so it's a very interesting snail, and it's mimicking kind of things that I look at in human cells, in old human cells. So these are the issues coming together. So I'm going to say, next slide, please. Okay, so I'm going to take you straight into the cell nucleus. So here we have a cell which is looked at analyzed by the electron microscope, but it's not this multicolored. This is pseudo-colored for attractiveness. But inside there, if you think of the cell like a fried egg, you've got the cell nucleus there is in that lovely sort of puce-colored with the darker regions inside that. So that's the cell nucleus. And what we're looking at there is quite a lot of DNA. Okay, so packaged into each cell nucleus is about 2 meters of DNA. And we have a nice cartoon here. From now on, take the human genome outside before you unravel it. Because yes, we've really got a lot of DNA that needs to be packed into this nucleus. And for the longest time, we thought it was a bit like spaghetti in a bowl. So that you would see it all sort of tangled up, sort of in this kind of situation. I'd like to say that in the 1980s, that we sort of, as a field, discovered that that's not the case. It's more like, if you want to say, meatballs or falafels or ravioli inside the cell nucleus. But actually, it was a rediscovery of somebody. There was a very famous microscopist called Bovary that already discovered this 100 years earlier. But the guy that was writing the papers at the time, Thomas Kramer in Germany, he is like, okay, this is a rediscovery. And then he wrote lots of nice reviews about the early work. So if we look here, the light color is more active chromatin that's opened, and the dark areas are chromatin or DNA that's tightly packed. So let's go to the next slide, please. And what you see here are chromosomes. This is called a karyotype. So these are the human chromosomes. 18 is ringed for a different reason. But you can see that they're different sizes. So we have a range of sizes of the chromosomes in their pairs. So this is a male genome because we've got an X and a Y there. They're different sizes. The pinched area, which hooks up to spindles when the cell is dividing, is called the centromere, and that's in different places in our on these chromosomes. And if you were to look at mouse chromosomes, they would all be this very similar size. They would be the centromeres would be in very similar places, and they would have very similar gene densities. But with our chromosomes in the human genome, they have different gene densities. So there are chromosomes that are very gene rich and chromosomes that are very gene poor. And so we have a range, a variation across our chromosomes. So the DNA is packaged up with proteins called histones, and it's wrapped around these histones, these proteins, and that helps to then compact the chromatin, what is then called chromatin. Once DNA is associated with protein, it becomes called chromatin, and then it tightens up again, okay, so it can be packed tightly into this nucleus. And there's an awful lot of organization and structures inside that nucleus that help that organization occur. So next slide, please. So when I finished my PhD, and I was looking at this was in Dundee in the very early 1990s, and I was looking at all the proteins, all the structure and scaffolding that you find inside the cell nucleus. But then I got a very nice postdoctoral position in Heidelberg in Germany with some of the guys that were actually inventing this process called chromosome painting. So then I didn't have to look just at the proteins, the structures in the nucleus, but then I could also look at the genome, because when I was doing my PhD, we would digest all the DNA and all the interesting chromosomal parts out of our cells when we were doing the preparations. But then I got to learn to do this painting, and that was with Peter Lichter and Thomas Kramer in Heidelberg. So we don't actually do it like this with little tiny paintbrushes painting individual chromosomes. That's not how it works. But we make a probe made out of the specific sequences that you find on those individual chromosomes. So you saw those individual chromosomes, and people have micro dissected them with very tiny needles, or they have flow sorted them so that they're in different sizes, and then you can make a specific DNA probe. And here we have some very beautiful pictures taken by a colleague of mine that I taught how to do fluorescence in situ hybridization, which we call FISH, which is the painting. And you can see these individual chromosomes. So these are in the interphase nuclei. So this is where the nuclei is active, it's replicating, it's transcribing its genes to make RNA that's going to go out and make protein. So they have very nice territorial regions that they sit in. So it's not like spaghetti at all, as we were thinking for a while. And you can see here where some of them are sitting right next to each other, here and here. You can see that they are not, there's a little bit of overlap, but not a lot. So they sit in their own specific addresses. So next slide, please. And what you can do once you've discovered that they sit in their own regions, are those locations random? Okay, so we know it's not like spaghetti, and we know it's more like meatballs or falafel, if you like, but are they then in just random locations? And they are not. Okay, so this was a big finding that happened at the end of the 90s. As I was in a laboratory in Edinburgh run by Professor Wendy Bickmore. And then we started mapping. So you can start mapping. And this is how we did it with FISH and with microscopy. You can do it in other ways now, sort of biochemically, and see which bits of the sequence is sitting next to each other. Because of the Human Genome Project, that can be done. But what we were doing was then mapping individual chromosomes. And this is a beautiful image here by Thomas Kramer, who's very famous for looking at the chromosome organization. And he, although this looks like a cartoon, he has actually, or people in his laboratory, should I say, have painted every single human chromosome a different color. So they're pseudo-colored. And so the computer will pick up signals from marker compounds and then color them all differently. But you can then map these. And what we have found is that in young cells that are actively dividing, they're organized according to gene density. So the chromosomes that are gene poor are at the edge of the nucleus here, this nice cartoon drawn by one of our undergraduates for me. And the gene-rich chromosomes are in the center. So this was a massive finding, and there's all sorts of theories around why this might happen. Perhaps it's a protection mechanism, okay, so that if you're getting assaults from outside, say radiation or something like that, or a toxin, then it's going to be the gene poor chromosomes at the edge that are most damaged, and then that will minimize the effect on those cells. Or that the mechanisms for making, for transcribing the genes, making so the RNA that goes to make the protein is in the inside of the nucleus. Next slide, please. But we then wanted to look at old cells. So always had an interest in then cells that then come to the end of their lifespan, if you like. They don't die. So when you have young cells that you grow in culture, if they're not immortalized, so they've not come from a cancer background, they will then only divide a certain number of times before they stop. They become what's known as replicatively senescent. And we have these senescent cells in our body. And as we get older and older, we have more of these senescent cells in our body. And they send out compounds outside of them that then affect other cells nearby them, that induce them to become senescent, and they start to destroy the matrix around them, so the collagen, etc., around in the tissues, and then this is why we get wrinkles and other organs age. So senescent cells are part of us, and they are part of the aging system that we go through. But when we looked at senescent cells, we found that the chromosome positioning had changed quite considerably. And these are where we painted chromosome territories. So these are the big chromosomes here going to the much more smaller chromosomes. And when we did the analysis, so it's statistically significant, lots and lots of nuclei looked at, we found that gene density wasn't the organization mechanism anymore. It was by size. And so you had the big chromosomes at the edge and the smaller chromosomes in the interior. And we can pick out senescent cells because we use a marker called Ki67. Here's in red. So this is a marker used for looking at cells that are actively dividing. And these are senescent cells. We know that because they are grown in high nutrients. They have plenty of space around them. There's no reason that they should then stop growing. Okay, and so they're in the right environment, but they don't have this marker. And here we have a nice picture of some senescent cells that normally when these cells are young, they're nice and long and thin. They don't have this strange morphology with all these fingers coming out of them. So next slide, please. So when we were looking at the younger cells, we found that, yes, you can then do things to them, shock them if you like. So one of the things that we would do is to take them into a non-proliferating state. Is similar to senescence, but not so, because they're young and they can come back into the proliferative cell cycle. So we take away their nutrients. So we take away the serum that we use to feed these cells, and that gives them, I guess, a bit of a shock. And what happens is we found that the chromosomes will then move to new locations. Okay, but there's the specific chromosomes that will move to the same site in the nucleus as they would do in senescent cells, but not everything. It's been very difficult over the years to find a really good marker to show something that is a young cell that's not proliferating at that particular point in time, but will come back into the cell cycle. And then a cell that is senescent, that's never going to come back into the cell cycle unless we use some of the new reversal drugs that I'll talk about later. Oh, sorry, can we go back? So here we have chromosome 10, and this chromosome is a fascinating chromosome because it sits in completely different locations. In proliferating cells, here, it's an intermediate location. And then quiescent cells, it's found at the edge of the nucleus. And in senescent cells, it's found in the interior. And I think this is something to do with silencing certain genes that are on chromosome 10 that are used for the cell cycle. But you want them reversibly silenced in quiescent cells, but irreversibly silenced in senescent cells. But that's something that we're looking into. So next slide, please. Thank you, Janet. So this, okay, so if you take the serum away and chromosomes are going to a new position, how is this happening? And how quick is it? I had a fabulous PhD student, Ishi Mehta, and we published a lot together from her thesis. I asked her to look how quickly it happens. So we started at 7 days and she worked backwards. So it's a big piece of work. I wish I told her to start at 5 minutes and work forward. But what happened was when we went back down, this happens, the movement of the chromosomes happens very rapidly. So it's already starting at 10 to 15 minutes. So I know these graphs are horrible, but here we have an intermediate location, because this is the edge of the nucleus here, and this is the interior. I don't want to go into how we do this, but we've been doing it for years and it works. So once you take that serum away, 10 minutes, chromosome 10 has started to move to the edge, and you can see it here by 30 minutes. Chromosomes 13 and 18 move towards the interior, and they end up being embedded and are so deeply associated with this structure called the nucleolus. Chromosome X is our control, never moves at all in these situations. So this is a very rapid relocation, and it's not random. These chromosomes aren't just going anywhere. You can do it reproducibly. And so this is a really nice mechanism. So it happens very quickly, it needs energy, and there must be a direction to it. Okay, so we started to think, how could this happen? Next slide, please. And okay, we believe that it's down to the sort of skeleton, the scaffolding within the nucleus that's not organized properly. But when we look at old cells, and we can do this by removing their nutrition, so we remove the serum from their culture, or we give them a mild heat shock. So here we have a heat shock situation down here. So this is chromosome 10. In senescent cells, you don't see this shift to the edge of the nucleus. Okay, in fact, it goes even more interior. Chromosome X pretty much does the same thing. Chromosome 11, that has heat shock protein genes on it, and we give it a mild heat shock, so we go up from 37 degrees to 42. If in the young proliferating cells, we get a movement, and those genes are moved, and another group have shown that they get upregulated and transcribed, so they're going to make the gene product. But in old cells, it doesn't happen. Next slide, please. So, as I said, we think it's to do with nuclear motors, and we're not the first people to sort of look at nuclear motors. So what we think nuclear motors are myosins and actin, and you may have heard of these proteins that are involved in our muscle movement. But we have isoforms of these inside the cell nucleus. So we have actin filaments that people can see. They're quite hard to see, but they have been seen by lots of different labs to have these little filaments. And we believe that the myosin, here's shown here, and these, so it's like these little tadpoles here, are binding to the DNA and then walking along these actin filaments and pulling DNA or chromosomes with them. And we can show it for genes, and we can show it for whole chromosomes. So if we inhibit nuclear myosin, the chromosome relocation mechanism doesn't work. And if you knock out the myosin, you can do it with something called siRNA, short interference RNA, where you interfere with the RNA, doesn't make the protein. It stops too, this movement stops. But when you look at the distribution of nuclear myosin in proliferating cells, this is how it looks like in the nucleus. You've got here big structures at the nucleolus, and then a homogeneous distribution with a rim around the outside. In quiescent cells, we don't need to, this is where we've taken serum away from young cells. It looks much more aggregated, but in senescent cells, this is how it has its distribution, and we've done the scoring, and that's pretty much all the cells look like that. So not like this at all. So am I running out of time? Sorry. Next slide, please. So the human summary is chromosomes sit in specific locations in cell nuclei. These locations are different for some chromosomes in old cells. Some chromosomes are rapidly moved to new non-random locations upon a stimulus. This relocation is not apparent in old senescent cells. Nuclear motor distribution in old cells is not as it is in young cells. Inhibition of the nuclear motor function stops chromosomes relocating. So next slide, please. Just run through quickly how the snails might be a good model for us looking at this. So this is our model snail, Biomiphalaria glabrata. It's had its genome sequenced, and we did the karyotype for this snail. And you can see that, again, we have different sizes of chromosomes. We have gene density is different, and the centromeres are in different places. And they also have these chromosome territories when you paint them. And this is how people in tropical regions get infected by this parasite. So next slide, please. When you have an infection and you use a live, what's called a miracidium, we get gene movement. And what happens with this gene movement from the edge to the interior, you get an upregulation and get the gene product. So we believe that the parasite is hijacking the snail's nucleus to do this. So what mechanism is the snail using to do this movement? Next slide, please. So if we use an inhibitor that we've used in the human cells, BDM, we stop this gene movement. So we can give the snails a mild heat shock, so they go from 28 degrees to 32, but we get a shift of the gene, an upregulation of the gene. We get gene products, which the parasite seems to use for its infectivity. But if you use BDM and block nuclear myosin, that relocation doesn't happen. Next slide, please. You can see that it's missing here inside the nucleus, where you see speckles here when we haven't used BDM. Next slide, please. So when we do this gene, look at this for this gene repositioning in old snails, we can't find it. Okay, so the genes do not move. So here's an old snail. So this is where the gene would sit normally. You then do a heat shock, nothing changes. But if you do a heat shock in young snails, 3 months juvenile, then we get this movement. Next slide, please. And we have much less myosin in old snails. Next slide, please. And so we want to use this snail as a new model for studying human aging. And we want to see if we can reinstate this chromosome on gene relocation mechanism, because we believe that that's important to ending up senescent. You can't move certain genes and transcribe them anymore. And so because the snail is in water and so amenable to being treated with different compounds, we're now going to look at a range of new anti-aging compounds and see what we can find out with the snail. So thank you very much for your time.
Thank you very much, Joanna.
Yeah.
That's really interesting, and really interesting research going on there in biosciences. That's great. So now we come to our final presentation with Ni Qin, who's going to talk about the Chinese migrants' low carbon lifestyle and practice have the potential to inspire more people in the UK. So when you're ready to share, Ni, that would be great.
Okay.
Look forward to your presentation. If you just want to say a little bit about your background or discipline as well, that'd be lovely. Thank you. Thank you very much.
Hello, everyone. I'm Jing. I come from Brunel Design School, and I'm the second-year PhD student. This is my topic today. The topic is about Chinese older migrants and the low carbon behavior of the Chinese older migrants in the UK. I changed my topic after 9 months review. Before 9 months review, I was always search some information and evidence about how to help the older migrants in UK. It means I treated them like vulnerable people. But after that, my supervisor gave me some inspiration. And she said, why you always think Chinese older migrants, they are vulnerable people? Why we can't learn something from them? And after that, I changed my topic like this. And after a long time, I searched more information. It gave me a lot of encouragement to continue this topic. As you can see, my topic is based on the global warming and net zero policy. Net zero means reducing greenhouse gas emissions to as close to zero as possible. And the UK has an aim to achieve the net zero emissions by 2050. I know these words at 2021. Before I didn't know it. I see the first time I know this word, I think it's so complete to me. What is net zero? But after I searched some information, I think we already did something to have the low carbon behavior and do something to about the net zero. If we divided our behavior or low carbon behavior into 3 levels, the first level is about the climate change and human behavior. It's more about global and about the world. Maybe we can't change it now. And the second level is about the human response and about the policy. This is what the government and the community can do. And for our individual, we can do is about our behavior change and get some support from the policy and the communities. The net zero and low carbon divided our behavior into 3 aspects. The first one is mobility. The second one is about home. And the third one is about food. But in the simple way, I think we can find some advice from the UN campaign. It gives us some very simple advice, like we need to save energy, we need to use more public transport, and we need to eat more vegetables, and we need to try to recycle and repair some devices or repair our clothes and something like that. After you read it, you will know we already did something like this, but maybe we don't know more knowledge about net zero and not more knowledge about low carbon behavior. For us younger people, we already don't know a lot of things about net zero. So imagine older people or migrants, they have language barrier, how they know this policy and how they know they already did some low carbon behavior. Why I choose the Chinese migrants as my user group? Because I searched some information and some evidence from the paper. It shows that the Chinese older people, they have meant to natural original value and collective value and something like, I can't see it, sorry. And morality values. Because I grew up in China, I already had this education before. My parents and my grandparents always tell me some stories like we need to save money, and we experienced natural disasters before, and we experienced war before. So we need to think about the future and save more energy, save more money for future. But imagine how Chinese older migrants think about it. They already maybe come here for 20 or 30 years or 10 years. Maybe they already changed their mindset after migrant here. And after that, I found some evidence about behavior change. Behavior change has 3 factors affected. One is internal variables, and the 2 is external variables, and the third is demographic factors. I use these models to guide my research and guide my questionnaire. The first one is the cultural adaptation theory, and the second one is low carbon behavior theory. This model gives me some inspirations about what factors I can add to enrich my questionnaire and make my questionnaire more wide and more suitable to ask questions for older migrant people. And after that, I did my first data collection research this research. And there are 4 channels I found my participants. The first one is I go to the Kingston Chinese Association to find some participants. And the 2 is I go to the Chinese church to involve their activity to find some participants. And the third and the 4th is from WeChat and Facebook. For older Chinese people, maybe the most useful app is WeChat, because they have a lot of groups in WeChat. So I found one older people to help me to aid me into this WeChat group, and I found some participants there. And total, I found about 33 participants. They come from mainland China, Hong Kong, Malaysia, and Taiwan and Singapore. But female, women more than men, maybe because women want to share more about their feelings and experience. The method I use is interview and questionnaire. I did an online interview and offline interview together. After that, I use Invivo and Excel to analyze my data. The aim of this research, the first aim is I want to help the Chinese older migrants to understand the low carbon behavior policy and the net zero policy, because some of them didn't know it and never think about they will know it. And the third goal is help me to understand their perspectives on the personal and social aspect of achieving net zero goals and understand their current practice. This practice involves 4 aspects. One is home, and the second is transport, and the third is about their clothes, and and the 4th is about how they treat the waste, how they treat waste. Maybe they reduce waste or they waste a lot. There are some initial findings from my first data collection research. The first one is about the generation difference. I think generation difference always talk about during the younger people and the older people, but during my research, the older people told me the different generations, they have different carbon emissions. One of the participants shared he has a son, but his son need to smoke and need to go to restaurants and need to buy some fashion clothes, but he doesn't need, because he retired at home and every day he just walk to the supermarket or walk to some place, and he even seldom use the public transport. So you can see the older people and the young people, they have different carbon emissions, and older people, they also seldom eat outside. Most of the time, the Chinese older migrants, they cook for themselves. And this initial finding, I want to talk about the one group people, they still remain low carbon behavior. It doesn't mean they don't have money or they are poor. After I interview them, I know some economic situation about their life. They already have enough salary to buy food and to buy everything, but they still remain the low carbon behavior. They give me some suggestions like we need to use public transport, we need to switch off the lights every day when we leave the home, and especially last year we have the energy crisis. They use the electricity blanket, and one of the older lady told me she even used the fire to get warm, but it's very dangerous. She didn't recommend us to do it. But they told me they didn't worry about the bills. It's because they always did this practice, and they always think about they need to save energy, they need to save money for future. That's why they last year they still continue this behavior, and maybe several years before, and before they come to UK, they always did this behavior. This I want to share some older people, they receive the benefits from the government. They change some behavior. It's something like that after they get the energy benefits from the government, and this is it is enough for their life, because if they live in the government's apartment, it's not very big. So the benefits from the government is enough. And even they told me they also left some money, but they don't want to return this money to the government. So maybe they will buy clothes, and they will go to beauty salon, and maybe they will buy food for their grandsons and granddaughters. So they share something like this. After this study, I think the benefits from government makes them change their behavior. But if they don't have the benefits, maybe they will still remain the low carbon behavior. This is talk about the international families. During my interview, maybe about 8 or 6 ladies, they married to local people, or married to Europe people, or their daughter married to local people, and some of them live together. They told me something about the Western culture and the Eastern culture. They need to suitable for each other. And in terms of eating habits, the female migrants will adapt to the eating habit for their husband and children. One older lady told me she needs to cook for herself because she wants to eat Chinese food, and she also needs to cook for her husband and children. Maybe her husband wants to eat some Western food, so it's a little complicated for her every day. And this is talk about energy crisis and COVID. Energy crisis, many of the older migrants talk about, all mentioned about energy crisis, but they also give us some suggestions of on how to save the energies. Like we can switch off the heating during the day, and we can wear more clothes when we're at home, and we can reduce the temperature at night, and we can also do some exercise to keep warm. And about the COVID, you can see the COVID emission has reduced the carbon emission in the last 3 years, and some participants said, I didn't buy a lot of clothes during this 3 years because I don't have enough social activities, and maybe some of their parents in hometown in China or in other country already passed away, so they reduced to go back to hometown to see their parents. And this is talk about the transport. Many of the older migrants, they told me they have the free transport card, so they can use this card for free to take bus. Some of the older migrants, they drive cars before, but after they get this car, they started to use the public transportation. But because of the strike, in the London, some participants also said it's not convenient to go outside or go to work, so they still need to drive cars. And especially they live in the countryside, they need to go to supermarket to buy some heavy stuffs. They also need to use cars. But they also said walking and by bicycle is also a kind of exercise. After retired, they stop, or they doesn't like to use a lot of flights to go to other countries or go traveling, because they think go to the airport is so tired and their body become more heavy day and day. So it's not convenient for them, and they don't need to hurry to go to the dissertation, to go to the place. So they don't need to hurry to go, so they can maybe use the train or use cars to drive to the place. And this is talk about the food change, the diet change. Some of the migrants say they already changed their eating habit because they move here for more than 20 years, and they try to cook and adapt the Western food. They try to eat cheese. They try to drink more milk, and especially I mentioned the international family. They need to cook. They need to diversity meals every day. And some older people, they also increase their consumption about meat and about the milk because they think milk and meat is not very expensive here. One old lady also shared with me, she used the milk to beauty her face, and when one old man shared with me, he even put the cheese into the dumplings. You know, the dumplings, we made the dumplings. We never use cheese into the dumplings. But the old man think, oh, cheese here is cheap, so I can use cheese to every dishes. Here is talk about food sources and vegetables. Many of the migrants, they don't like the imported food because they think it's not fresh and is more expensive than local supermarkets. They prefer to buy vegetables and fruit, fresh vegetables in the local supermarket, and they said they must eat the vegetables every day. You can see this table here. 26 participants said they eat vegetables every day. This is talk about reduce waste. This is the same with the eat vegetables. Many participants said they bring bags to the supermarket, and some older lady shared with me they change their old clothes into bag, and they also use some waste paper to make notes and make their own filter lights to buy expired food, so they never waste the expired food. This is talk about clothes. This data is a little bit interesting because I interviewed most of the participants are women. 23 participants are women, and many of them mentioned that they really like to buy clothes before, or they have more than 100 or 200 clothes in their wardrobes. But after that, they told me because they get old, they stop or they reduce to buy more clothes, and they never waste these clothes. Some of them donate these clothes to the charity, and some of them send the clothes to their friends in China, or sent to their daughter or granddaughter. So they didn't waste the clothes. But the data shows that they already spent a lot of money or emissions on the clothes. And they told me, especially in UK, the clothes are very fashion and not very expensive. For them, maybe it's very easy to buy. This is talk about the barrier to access the low carbon behavior. Before the older Chinese migrants, they didn't know a lot of things about net zero or low carbon behavior. After I introduced the low carbon behavior and net zero policy to them, and they told me it's not very convenient, or they don't interested in it. And I summarized the barrier, what they face. The first one is they have the life circle, the limited life circle, especially some older Chinese people, they don't have high education. They prefer to talk with Chinese people and prefer to go to the community for activities. So they really get some news from outside. That's why they don't know a lot. And the second is about their personality. They are so shy and they don't want to talk to strangers, and they are afraid to know more knowledge here, or especially their language is not good. They are scared of English. And the third is they think the news here, they can't trust it, because different newspaper, different TVs report different news. They don't know what they can trust. They asked me to provide some official documents to them so they can trust it. And the most important is language barrier. Because of the language barrier, many of the migrants, they can't travel, or they can't go outside, or they can't communicate to local people. And the net zero policy, they're all English and they don't have Chinese version. So many of Chinese people, they don't know it. And the last one is about their eyes and their body. If they get more old, they can't go outside. And if their eyes are get bad, they can't read or can't read news, can't watch TV. That's all the problem. They didn't get enough low carbon information. I have a short discussion. If we talk about the personal lifestyle, and if we want to change the lifestyle of older people, we need to consider many factors. We can't just say, oh, I give you a policy and you just follow these rules and you can know what is low carbon and you will have low carbon behavior. We also need to consider about our social environment, about our culture, and we need to provide enough support to them and let them know what they need to do or what they can do. If we can help like Chinese older migrants, we also can help more people in UK or in London. Maybe it's more easier to help younger generation like this. That's what these pictures show, that if personal lifestyle change, the social environments will change. And if the social environment change, it also affect our lifestyle, and a new personal lifestyle will come out. Thank you.
Thank you, Quinn. That was really interesting. Thank you very much.